Welcome to Summarize Science. Have you ever wondered why, with all our advanced DNA sequencing technology, the discovery of new genes linked to diseases seems to have slowed down? It's a puzzling question that scientists are tackling right now. This paper, Advancing Risk Gene Discovery Across the Allele Frequency Spectrum, dives deep into this problem and offers a fascinating answer. For the last 2 decades, geneticists have gotten really good at finding 2 kinds of genetic culprits for diseases. First, there are the rare sledgehammer mutations, single, powerful changes in your DNA that have a big effect. Then there are the common feather variants, tiny, subtle changes that millions of people have, where each one only adds a tiny bit of risk. You can see the slowdown in new discoveries in this chart, where the rate of finding new gene disease links has dropped dramatically. The researchers argue this isn't because we've found them all, but because our methods have hit a wall. This second chart reveals something strange, a surprising pileup of variants classified as benign or harmless in a specific frequency range. The authors believe this is evidence of a huge genetic blind spot, which they call the missing middle. These are variants that are not super rare, but not super common either, and they have a moderate effect size. Our current tools are either designed for the sledgehammers or the feathers, and they systematically miss these in-between variants. So, that pileup of harmless variants isn't real. It's just a reflection of our inability to detect their moderate effects. So, how do we find what's hidden in this blind spot? This diagram illustrates the challenge and the path forward. Unlike a single rare variant that can push you over a disease threshold by itself, these intermediate variants require a new way of thinking. One new strategy is to look for combinations of risk factors. Instead of looking for a single knockout punch, scientists are searching for a 1-2 punch, where 2 or more moderate-risk genes together create a problem. This has been used in cancer research, where multiple mutations are often needed for a tumor to grow. Another approach is to stop thinking about diseases as a simple yes or no diagnosis. Many conditions, like autism or high blood pressure, exist on a spectrum. By studying these traits as continuous measurements instead of a binary label, we gain a massive amount of statistical power to detect more subtle genetic effects. Finally, scientists are using network analysis. This involves leveraging what we already know about how genes and proteins work together in complex biological networks. If a new candidate gene is heavily connected to genes we already know are involved in a disease, it's a very strong clue that it might be important too. So let's recap the key points. The slowdown in gene discovery isn't the end of the story. It's a sign that we need better tools. There is a missing middle of medium-impact genetic variants that our current methods are blind to, and this is where a large chunk of heritability for complex diseases likely lies. To find them, researchers are developing new strategies, like looking for combinations of genes, studying traits on a spectrum, and using network biology. By filling in this blind spot, we can get a much more complete picture of the genetic architecture of disease. This could unlock new diagnostic tools and therapeutic targets for some of the most challenging health conditions we face.